This science report was asked for by Marilyn because it's come up on a number of EAB meetings that there's interest in looking at endophenotypes and biomarkers with genetic analysis, which of course all of you do as well. But this is designed to be a little bit of an overview of what's going on in that area. And there's also been contributions from Claudia Sitazabal, who many of you know, Paul Crane and Olivier Lichtage, both to the written report, which is on NIAGADS, and to this presentation today. So how do we find, oh, sorry, before I proceed, Anita, do you see this full screen just so everyone can see it?
I do, yes.
Okay, wonderful. So how do we find genetic variants that help or harm the brain? I mean, the idea that brain measures, and for that matter, fluid biomarkers or other biological measures may be a good target for genetic analysis comes from a few sources. I mean, one is disentangling the biology of the disorder that we're studying. Obviously, if there's a precise biomarker, whether it be in an image or plasma, CSF, of a biological process, it's more specific if we find genetic or other determinants of that process. The other argument, which was made early in this type of work, was that it might be more efficient. And so certainly in biological psychiatry, where the Psychiatric Genomics Consortium was requiring 50 to 100,000 people for their GWAS, there was a thought that maybe the genetic architecture might be simpler, or genetic effects might be stronger on refined measures of the biology. And even if you don't believe that, there was some interest in revealing how susceptibility loci exert their effects in the brain, or not. I mean, they could certainly work via other biological pathways. So I think the arguments for doing this and screening biomarkers and images have come from a number of sources. Now, some early skeptics of this included Nick Martin, who's the president of the Behavioral Genetics Association, and he had said, well, if you want to discover genes that affect the brain or genetic variants associated with brain metrics, you'll need brain images from 50,000 people. Just because images are more expensive to collect than measures of height or weight, it doesn't change the power calculations. Now, you all know that that's not quite correct, but we'll look into it in more detail. So this motivated efforts from the ENIGMA Consortium, the Charge Consortium, many others collecting images to pull together, much like the ADG was, metrics derived from brain scans. Here they're subcortical volumes on MRI, and do a genome-wide association with a quantitative trait. I mean, you can see this paper in Nature had many hits, different hits for different structures. So I'll tell you a little bit about the background and how this is done and how it's going. So ENIGMA, which is a consortium, it stands for Enhancing Neuroimaging Genetics Through Meta-Analysis, was founded 14 years ago and has published the largest neuroimaging studies of now 15 brain disorders. Now, this is not exclusively a genetics consortium. The goal is to discover factors that help or harm the brain, but in the course of doing so, we've analyzed brain scans and genomic data, whole genome data, genome-wide association data from over 100,000 people. Many of the brain GWAS conducted in close partnership with Charge and other ADSP members over the last 12 years. I'll explain that in a moment. But this has involved work with people from 45 countries and 500 medical centers contributing data, much like the AD GWAS, contributing image-derived data as well. And this has also led to the largest neuroimaging studies of epilepsy, Parkinson's, ataxia, brain injury, and all these psychiatric and neurodevelopmental conditions that you see there. So certainly the imaging workflows are very well validated. So just to speak of ENIGMA for a moment, this analysis of the brain images had two main goals. One was not genetic. It was to understand how over 30 major disorders affect the brain. MRI, diffusion, functional imaging, EEG, and MEG are being used to evaluate that. But then secondly, to evaluate how genetic variation is associated with brain metrics, either with GWAS or epigenome-wide assessments of methylation or CNV analysis, the very active group in ENIGMA looking at structural variants, to find genetic markers and of course, environmental factors that might affect the brain's development or aging or disease risk or treatment response. Now, I will say that most of these GWAS studies have been partnerships with Charge. Now, Sudha is on the call, and so is Josh Beers. So I want to give a shout out to Sudha, Josh, and Claudia Sitazabal, who's first authored many of these, Charlie DiCarli, and many ADSP members who've led, these are certainly by no means all ENIGMA studies, they've led many of the analyses that I'll talk about. But the ENIGMA data in particular is spread out. It comes from all over the world, and it's often analyzed locally. So much like AD-GWAS, there will be a meta-analysis typically of locally run GWAS. And of course, the results are weighted by the sample size that each group is providing. So within ENIGMA, there are all these different groups focused on psychiatry, neurology, brain development. And there's been workflows, of course, to look at case control differences. So if you were to argue, well, how do we know that any of these brain measures have anything to do with ADRD or particular disorders? I mean, they certainly do, which is why these, in this case, cortical thickness measures were chosen as one of the biological targets for GWAS. Now, all of the data has to be harmonized. I see Mike Kukuro is on the call. I mean, he and others are harmonizing ADSP data. I mean, obviously from around the world, both the imaging on the left and the genetic data on the right have been harmonized by groups that are, as ADSP is very, very expert in doing this. So some of the key brain GWAS you should know about. I don't want to neglect the biomarker GWAS, the CSF and plasma GWAS that are very important as well. I'll mention them later. But the brain GWAS have included analysis of brain volumes on MRI for Alzheimer's and related dementias. Certainly hippocampal volume is interesting. So are other subcortical volumes, cortical thickness and surface area. This now has over 500 genome-wide significant hits. There was a paper led by Katrina Grasby and Neda Jahanshad in Science, and one by Edith Hofer and the Charge group. And I will say that the partnership between Charge and ENIGMA and other consortia has led to an ability to replicate many of these findings. So I mean, it's been immensely supportive. There's a new GWAS on rates of change of brain volume. So if you were to measure the interval change in hippocampal volume or other volumes on MRI, you could ask the question, are there any loci associated with that? Now that GWAS has over 10,000 people, not quite as many as the first one, but it already has 15 hits. There's a great study many of you took part in. Last author is Carlos Crishaga on amyloid PET. I'll tell you about that later. Charges analysis, I see Miriam Fanage and others are on the call. They've been leading analyses of vascular disease and white matter hyperintensities derived from imaging. And I won't talk about them today, but there's also GWAS of brain function with EEG and task-based functional MRI, remarkable work that's really bearing fruit finally in ENIGMA, led by the Charité group in Berlin. Now, of course, motivating this is to understand the genetic overlap between all of these brain GWAS and other AD or Parkinson's or other dementia GWAS. And I'll tell you about some of the other ongoing studies as well. It's not all GWAS. There's certainly epigenome-wide association analysis, structural variant analysis, looking at CNVs in the brain. And what we will call AI-enhanced GWAS. Many of you are familiar with doing GWAS on proxy variables, ones that are easier to collect or more plentiful, or in the case of AI, ones that might be better tuned to efficiently finding hits. And I'll talk a little bit about proxy phenotypes and dementia subtypes, because there's work funded by ADSP in the AI group that is disentangling dementia subtypes and then going ahead and providing scores to genetic analysis. So here's what you do. I feel very humble speaking to this group on this subject. But for people involved in brain imaging, they're not always familiar with GWAS. But of course, after imputation, which might be to TopMed or in the past 1,000 genomes, you can go ahead and regress the allelic dose at each locus against the quantitative measure from the brain. Here it's intracranial volume. And you can fit, as you know, dominant, additive, and other models. But this is mass univariate. This is distributed. So what ENIGMA does, and I understand what Charge does, is very similar, is a protocol is circulated for quantification and QC of data. It could be structural MRI. On the right, you see diffusion, tensor imaging. I mean, these are all workflows that are widely used in labs internationally. And then after imputation, you would go ahead, do GWAS. You can do other types of analysis as well. It doesn't have to be genetic, but in the end, each site would centralize their summary statistics, and you'd meta-analyze them, much like Brian and others here do for the AD GWAS. So what do the measures look like? Well, they're age dependent. And so if you look at thalamic volume, or for that matter, most brain measures throughout life, they have a U-shaped trajectory with a rather precipitous decline after age 60. So there are also age effects fit, and for that matter, post hoc tests of age interactions with SNPs. Because, of course, they may influence development, but not aging, or conversely may come into play late in life. And there's some evidence that the ApoE haplotype increases in its influence later in life, which we'll show in a minute. So this is hippocampal volume. I mean, many of you are familiar with this. This is the ENIGMA Lifespan Project. There are many other projects like this. There's one called Brain Charts, led by Richard Bethlehem, one led by Christos Stavratsikos, called Eye Staging. There are many others that aggregate phenotype data like this. And you can essentially fit models without genetics and say, some structures of the brain, here the subcortical structures, have a rather modest decline. Look at the amygdala until age 45 or so. And others have different trajectories. Of course, that one at the top left with thalamic gain late in life, that's just a selection bias, where if you're healthy enough to come to the scanner, you probably have a rather protected brain. So the second ENIGMA charge GWAS of hippocampal volume, many of you on this call contributed. I see Josh and Sudha and others are on this call, yielded 6 loci, 4 of them novel for hippocampal volume across the lifespan. So these are studies that go from young childhood through to old age. This is one with 33,000 MRIs. Now, of course, the benefit of brain mapping is you can then plot the allelic variation back into the image using statistical parametric mapping and see, I mean, that, believe it or not, is the hippocampus. You can see exactly where the SNP variation is associated with, in this case, shape variation in the anterior hippocampus. One thing that the ApoE experts will see is that in the genome-wide analysis that aggregated data throughout life, look on chromosome 19. So ApoE did not come out in this, and it's a 30,000 scan analysis. The main reason being, and here you will see it, is that the ApoE effect, at least on hippocampal volume, is age dependent. And so by the time, and I'm making a lot of simplifications here about ancestry and other things, but by the time you're 70 or 80, one copy of ApoE4 will shift your hippocampal volume by 1 or 2 standard deviations. But when you're 20 or 30, and there's many cohorts even in infants that have looked at ApoE4, at least on average, it's rather modest. So if you do a GWAS and you include younger people, you won't always find all of the famous AD risk genes, which is a motivation for stratifying your GWAS, obviously. So anyway, the first thing to note, this is analysis by Derek Hybar at the top, and here Adams working with the Charge Consortium and ENIGMA on the bottom. Intracranial volume at the bottom has a number of hits. Hippocampal volume at the top has a number of hits. One interesting one is the MAPT gene. There is an H1H2 inversion. It's very famous in the vicinity of that gene that's very strongly associated with dementias. I mean, it's a really interesting area. It is also a common variant SNP in LD with that inversion is associated with the total volume of the brain, which is quite interesting. Not in an expected way. So the risk conferring variant for PD, this is rather an old Parkinson's GWAS. I apologize to Andy Singleton, but his Mike Knowles is old GWAS. You'll see MAPT over there as the second leading hit. Synuclein alpha has a SNP, that's the lead one over there. But the variant that confers increased risk for PD actually increases or is associated with an increase in intracranial volume earlier in life. So there's all sorts of theories about whether that's inflammatory or an early protective effect. Alan Daguerre has written at length on this subject about whether there's an antagonistic pleiotropy in which a developmental advantage is conferred by that marker. Anyway, then this paper led by Claudia Satizabal, I think she was at BU at the time, she's now at the Glenn Biggs Institute with Dr. Sudha Seshadri and others. This remarkable paper pooled ENIGMA charge and UK Biobank data. By then, the UK was scanning pretty much everyone in the UK. If you want a lot of MRI scans, there are 46,000 available from UK Biobank. So this greatly empowered the studies, and you'll see those structures there on the top left are the subcortical structures. I don't know if you would recognize them, but there's caudate, brainstem, amygdala, many others. And there have also been structure-specific GWAS that have confirmed many of these results. And just a latest update, because there's supposed to be an update on the latest. There's a rather brilliant analysis by ENIGMA Charge UK Biobank, and also a new imaging cohort, ABCD, which is 10,000 adolescents. They're rather a diverse sample, perhaps more so than many of the others. But adding 10,000 adolescents to this has really helped. Now, you're all very familiar with this. If a genomic locus had predictive value late in life, but not in childhood, it would not necessarily come up here. So these are just loci that are associated with these volumes. 51 hits for the hippocampus. But if we age stratify these, you may get different ones or others. And I mean, there's more detail is provided in this preprint. Now, you could ask, does any polygenic score prorated from these GWAS explain anything at all about the imaging? And it does. So this is the fraction of the phenotypic variance explained by a PRS score that comes from the plots that I just showed. So, I mean, you don't need reminding of this, but if you took all the loci that passed genome-wide significance threshold or some other threshold that is empirically chosen, you can then go into independent data, which wasn't used for the discovery GWAS, and ask, look, does this PRS, or polygenic score, I should say, account for much of the volume variation? For intracranial volume, it's pretty good. I mean, that's 9 or 10%. For hippocampal volume, it's about 4 or 5. And then you have the ancestry specific GWAS. You'll see a drop in the amount of variance explained to about a half of that in non-white Hispanic individuals for a number of reasons. I mean, both the differences in genetic architecture, possibly, although there are not strongly confirmed loci that are ancestry specific for brain. And then secondly, the lower sample size, as you know, for the non-white Hispanic cohort. So, I mean, obviously, if that was fixed, many of these results may be improved. Now, is there any increased efficiency in doing brain, or for that matter, biomarker GWAS versus case control GWAS? Now, that's not the only reason to do it. I mean, we're not doing it just because it's faster. I mean, there is some analytic breakdown of the processes that can be achieved using biomarkers. And then as a flip side, we also would very much need AD GWAS, and it would not be important to find a locus that influences the brain if it had no bearing on disease. So just to sort of look in context, a very nice paper by Anders Dale, last author, and Dominic Holland from UCSD and their colleagues in Norway, where they've ranked many of our favorite clinical phenotypes and endophenotypes. There you see AD in green. And on the x-axis is how many subjects. I think they pulled both cases and controls. Do you need? This is a little bit old, as you know from Brian, your work and others, the AD GWAS has a higher sample size right now. But on the y-axis is the proportion of the SNP-related tag variants that has been accounted for by the GWAS. Way over on the right, there's phenotypes like major depression that have been not entirely fruitful until recently for GWAS. And then biomarkers that have a very simple architecture, HDL, for example, is a favorite one. There's a lot of simplification here for those of you who are quantitative geneticists. I mean, they fitted a sigmoid. It's not necessarily the case that the variants come in this smooth arc, but the rank order is reasonable. I think at the time, the motivation to do this was just to show that some phenotypes may be more efficient, particularly if they're a proxy for another one that is harder to collect. Now, one thing that you will want to know is, do any of these GWAS bear any relations to any of the others? So this paper by Katrina Graspin, Eda Jahanshad, ENIGMA, and Charge and UK Biobank in Science in 2020 showed that if you steer around the cortex and you ask which variants are associated with, in this case, the surface area of different regions, you get both common and distinct loci, as you'd expect. And then, I mean, here's the extraordinary paper about this. And if you go ahead, and I mean, here's all the loci that affect anything measured in the cortex, and you see if there are any clusters. There are not particularly any clusters in the genome. I mean, here's a 3D Manhattan plot where the brain traits are arrayed along one axis, and the usual genomic axis is shown, and the p-values are there. There's some extraordinary SNP that influences the cortex in the vicinity of the sensory motor homunculus, which is quite interesting, which is also conserved across species. But we try and do this properly and show you it with a bit more insight. If you look at the genetic correlation matrix, and you can run LD score regression on the summary stats from these ROIs, you will see a block-like structure. I mean, there is some evidence that there are unitary sectors of the cortex that have common genetic determination, and then still others, and they don't particularly coincide with neuroanatomic atlases. So, I mean, for those of you interested in doing brain GWAS, I mean, somehow, and this has been proposed for many years, somehow aggregating these phenotypes in a multivariate way might be more efficient. And there are papers by Dennis van der Meer and Ole Andreas and many others, probably most of you here, that have exploited this genetic correlation across phenotypes to improve the statistical power. Okay, so do any of the loci that influence the brain have any pleiotropy or genetic overlap or concordance with those that have been found for Parkinson's disease, depression, ADHD? They certainly do. So AD is very complicated because the predominant common haplotype influencing late onset AD risk, APOE, has this very strong age dependency in its effect on the brain. So you will see papers showing rather modest overlap, but if you look late in life, it's very strong, mainly driven by APOE, but there are other brain AD risk loci with brain-related evidence, not just from MRI, but also in psychiatry. Many of the psychiatric GWAS, particularly insomnia, which is quite interesting, show a negative pleiotropy where loci associated with larger volumes, I mean, things that we would assume are beneficial, but that's not entirely always the case. They tend to be associated with less risk of the disorders shown here, with the exception of Parkinson's, which is a complete anomaly because the MAPT variant that increases PD risk also increases cortical surface area. And it is, I understand, the lead SNP in the intracranial volume GWAS, so there's something interesting going on with tau. Now, if you don't want to download all of this, you can go to the ENIGMA 3 browser. It's on the internet. You can look up SNPs and find phenotypes that they have been associated with. We try and keep this up to date. The cortical GWAS is in there. And you can download Claudia and Sudha and Josh's charged GWAS from here. They've very kindly allowed us to release those from our website. The procedure to access them, it does acquire very brief approval, but you're very welcome to download and use all of these brain GWAS in your genomic analyses. Now, I'd be remiss if I just stuck with anatomical MRI. Charge has been looking also at component processes in dementia that are evident on brain imaging. This one looks at evidence of cerebrovascular disease. Many analyses in Charge have been looking at this. What is quite interesting about it, this is work with lead author Paul Nyquist, is that if you were to segregate the white matter hyperintensities by region of the brain into periventricular and deep white matter hyperintensities, and this is in data pulled from Charge, a little bit of data from ENIGMA, not much, and UK Biobank, you'll see partially distinct sets of genetic loci, which I think motivates the deep phenotyping in the imaging. I mean, if there is a regional segregation to the genetics, and the image at the top there, I borrowed it from Maika Vanui at Erasmus. She's given excellent talks on this subject as to why the genomics of even component processes in dementia might be regionally segregated in regard to their genetics. Now, I don't want to stick with MRI. So those of you working in the ATN framework, or there are many biomarkers in imaging that look directly at tau or amyloid, or in this case, amyloid PET. This very nice study came out in April. Last author, Carlos Crishaga, many of you are contributing to this one, showing a very interesting genetic architecture for amyloid deposition measured with PET in just over 13,000 individuals. First of all, the markers do overlap with clinically ascertained AD, FTD, stroke risk loci, and there are many novel ones which are extremely interesting as pharmaceutical targets. There is also an ancestry dependence, which many of you have discussed on these calls and at AAIC. Certainly, there's a lot of interest that there are 4 independent loci in the ApoE region, some of which show an ancestry dependent effect. I mean, all of you are the experts on this, and you've gone through this in great detail. There is evidence that the ApoE4 effect on amyloid is a little bit weaker in Asian ancestry individuals than in non-Hispanic whites and African American or African ancestry individuals. ApoE2, at least in regard to amyloid, appeared protective across all ethnicities. But the paper is very interesting to read on this. I don't want to prejudice your interpretation of it, but it looks like there's a hint of evidence for an ancestry-dependent protective effect on amyloid accumulation from PET. Now there is this is just out, so this also is work by many of you looking at fluid biomarkers, measures of A beta and p-tau. This is just last month. I strongly encourage you to read this if you're not an author of it. It's 31 cohorts with the rather extraordinary sample here for CSF, A beta, and tau. ApoE comes out strongly. Other favorite AD susceptibility loci in CR1 and BIN2 come out. CR1 for A42, BIN1 for p-tau. And a couple of new ones that both associated with p-tau in CSF and lateral ventricular volume, implying that there's very nice analysis implying that there is a correlated effect in a brain measure that can be imaged. Now, just in the last, maybe we'll carry on for 5 minutes and then we'll leave 10 minutes for commentary. There's a great deal of interest in brain genetics and structural variants. And so parallel to this, Ida Sonderby and others in Norway as part of ENIGMA have been cataloging rare duplications and deletions in the genome. As you are in ADSP, this is with particular interest in neurodevelopmental conditions. Many of you know that there are deletions that enhance or cause risk for many conditions, autism, schizophrenia, many others. So they've been cataloging them. Now, if you finish a GWAS, you can go and call CNVs. And even if they're very rare, as many of these are, you might have enough to look at the brain effects of duplications and deletions. The most famous is perhaps the 22q11.
Where did you go?
Oh, sorry. Is there a question? Okay. So just to sort of showcase what is going on here, many of these genetic deletions or duplications are associated with very large effect sizes in terms of the effects on structural measures. These are surface area measures, cortical thickness measures. To the neurodevelopmental geneticist, this won't come as any surprise because of the profound behavioral effects. So there's some interest if the idiopathic disorders that these genomic loci confer risk for have a similar pattern in the brain. The answer seems to be not always. And so this is very wonderful work by Carrie Bearden, Sebastian Jacquemont, and Clara Moreau, who's now a postdoc with us at USC. And they're compiling these, as a reference. It'd be quite interesting to look at this in AD. So maybe at the end we could talk about that because there's some convergence between this project and some of the structural variant projects going on. Now, I won't tell you about EEG other than to say it's very convenient to do. And there's a very nice story. So the lead SNPs for power spectra in EEG are implicating genetic loci in the GABA-A signaling pathway. And it's really interesting. So this is in a genome-wide analysis of multi-cohort EEG data. The GABA2 gene has a SNP receptor subunit of the GABA-A neurotransmitter that not only influences risk for alcoholism and epilepsy, it was very unclear why, but it also directly affects spectral power. So this is very exciting. I mean, if some genomic markers affect functional brain synchrony and they overlap with some of the psychiatric loci, that could be quite an interesting discovery. Methylation is interesting too. Now, there's not quite as much data with imaging and methylation, but this work by Tianya Jha working with ENIGMA Epigenetics And Sylvain de Rivière, here she is shown, she's at King's College London, has found that methylation of HOX genes, homeobox genes, which are critical in development, predicts regional brain volumes. And so this, I mean, it's hard to neglect if it has this much of an effect. And so I would really recommend looking at their work, that DNA methylation at age 14 not only predicts, and I say predicts in the weak sense, I mean, it's just associated, but it is associated with future onset of a very large range of mental health symptoms. She's primarily interested in adolescent mental health, but it'd be very interesting to look at these genes and their methylation status later in life. And there's a list. This is unpublished, but it's very, very, very cool indeed. Are there variants that affect the rate of atrophy? Now, you could say, well, the structure of the brain and its genetic correlates may be completely irrelevant to dementia if all of the effects happened early in life. And so this remarkable paper by Hilke Holzhoff-Paul, Rachel Brower, and ENIGMA's plasticity working group measured in 10,000 people the rate of brain change. Now, lateral ventricles get bigger, nearly everything else gets smaller. This is the derivative, it's the time derivative. So I showed you earlier plots of the volumes of these things. This is the rate of change of these things. And you can do a GWAS on the rates, and you'll find, or they found, in a nicely replicated design, 15 loci that were associated with the rate of brain atrophy in different regions. And some were age dependent and some were not. So I mean, I'm sure they were all age dependent, but the interaction was detected with age in a number of these. And so this would be very interesting to overlay with some of the emerging loci coming from ADSP. And anyone's more than welcome to get this if you want to add it as a sentence or a paragraph to your outgoing genetic papers. Okay, what about mathematics? So you don't have to do mass univariate genetics. You can do multivariate analysis in the image. You can find a pattern in the image, or you can find a polygenic risk or a linear combination of loci, or both at the same time. You can do deep sparse CCA. Li Shen, who's on the call, and his team have pioneered deep sparse CCA to go pull out from the image and from the genome optimum, they don't even have to be linear combinations, optimum functions of the genomic and imaging data to lock onto important markers. Now, this isn't with total disregard to systems biology. They also, in that review paper, there's a really nice review paper, which I co-authored with Li Shen, although he actually wrote it, that integrates systems biology and Go pathway analysis and many other sources of information as constraints and guidance for these discoveries. Another line of activity led by Christos Davatsikos and to some extent by De Guigi, who's one of the AI/ML researchers, is to try and find dimensions in the imaging data. You've seen this for PET in Jake Vogel's work and others' work, where there are dimensions of evolution and subtypes and stages of different biomarkers. And if, for example, there were subtypes, and we know that there are, it would be more sensible, rather than to agglomerate them all, to give each individual a score for the presence or expression of each subtype, and then go ahead and do GWAS on the score. So this is really wonderful, and there's starting to be enough data to do this in terms of frontal predominant or limbic predominant atrophy, and for that matter, PET. And you can either do this with CCA, or you can do other types of subtyping and staging using an algorithm called sustain. The other thing that AI can bring to the table, and this is work by Andy Sakin and Taiho Zhou and Kwang-Sik No, who's on the call, is you can go run a deep learning convolutional neural network and find the PET signatures or MRI signatures that are most tightly associated with AD. And then you can run that network on tens of thousands of images and use its opinion as a proxy, and then run GWAS. So this notion of sort of AD by proxy, as you know, is quite popular. I mean, you look at family inheritance of AD rather than ascertained AD. So rather than bother to phenotype huge numbers of images, and the court is out on this one. It's not a panacea by any means, but you would certainly try. And in work by Taiho Zhou and the Indiana group, I mean, this has been proven actually very, very efficient because it'll lock onto not just PET phenotypes that are interesting, but ones that are in a certain anatomical pattern, and it'll get rid of all of the other noise that isn't important to disease progression. And then last of all, but I won't dwell on it, you can do GWAS on these things, and there is underway in ENIGMA and CHARGE a broad range of measures of brain connectivity being subjected to genetic analysis, but we'll save that for another day. So thanks to NIA and ADSP and the Alzheimer's Association and all the other federal and private agencies worldwide that funded this. And a big shout out to Dr. Sudha Seshadri, Dr. Claudia Satisabel, and Dr. Josh Bish, because even though I've presented this as the PI of ENIGMA, I mean, you all know that the same as with ADSP, many of the papers I presented were led by them or originated by them conceptually and in their execution. So thanks again for the invitation, and thanks, Anita, for hosting this call today.